In this video, we will discuss about ovarian clear cell carcinoma or OCCC. And in this disease, the PPP2R1A mutations will cause increased efficiencies of this ICB, that is immune checkpoint blockade treatment. And our research will be based on this hypothesis or these outcomes that the researchers are able to finally come up in this research paper. So how they did their studies? So they take their patients who are affected with this OCCC, that is ovarian clear cell carcinoma. They take around 34 patients and do the analysis pre-treatment and the on treatment, and they are getting treatment with this ICP. ICB is immune checkpoint blockade therapy. That is, it will block the suppressive or checkpoint on the immune cells that helps in the suppressions of the immune response through this anti-CLA4 and anti-PD or anti-PD1 or anti-PDL1. And then what they did is they analyze the survival probability of the patients with different conditions and did the mutational landscape and the transcriptional profile, and also did the proteomics profile, functional validations, and then external validations. This is in brief how they conduct their studies. So in the panel B, they shows the statistical analysis of some of the patients that they have analyzed and what are the conditions that they are undergone clinical trials and whether they undergoes both the combinations, the treatment arm, that is both the combinations or the sequential combinations. And thereafter they also checked whether they have these mutations, that is ARLD1A mutations is there in the patients or not, and with different time course they check or what are the treatment they have undergone through, either anti-PD1 treatment or this ongoing follow-up is the final step, or the SD treatment, PR or CR treatments. And first what they are able to find out from their this analysis is that PPP2R1A mutant patients will have longer survival rate. In order to validate those hypotheses, they took out the patients who are affected with this PPP2R1A, that is the patients that have the mutation variant of this PPP2R1A and patients that have the wild type variations, and then they check the survival probability. They are able to find out that survival rate is higher in the case where patients are affected with the patients have this mutated PPP2R1A, and then they check that within this, apart from this mutant, the ARLD1A mutant also cause increased survival rate. So they independently checked they did the similar exercise, that they checked the patients with AR1DA1 wild type and AR1DA1A mutant, and they are able to see that patients that have this AR1DA1 mutant have a higher survival rate compared to the case where patient does not have that mutant. So once they find out that these two mutants has the highest significant impact on the survival rate, next what they did is they check the ARLDA1 subset populations in the ARLDA1 mutant subset populations in the PPP2R1A mutant populations, and they are able to see that these PPP2R1A mutations will still has the highest survival rate compared to the case of this where PPP2R1A mutant are present. And next they show some of the topograms of the patients who are affected with the tumors, and the arrow shows the tumors site. We can see that at the baseline level they can see there are some tumors levels, and after 12 weeks of treatment they see some increase in the tumors, and after 24 weeks the tumors is significantly gone, and after 35 weeks also the tumors doesn't seems to come up. And when they check the percentage change from the baseline of the tumors, we can see that in the case of where the PPP2R1A knockdown and AKT knockdown is there, PPP2R1A and AKT wild type is there, in that case we can see the tumor is increasing, whereas when the AKT mutation is there, we can see the tumor is decreasing. And when the PPP2R1A mutations are there, we can also see in that case the percentage baseline is somewhat decreasing compared to the case where PPP2R1A present in the wild type. And here is the statistical analysis of almost the 100 patients, which is shrink. And here we can see that the patients who have the wild type PPP2R1A and AKT wild type has gone up in the percentage change in the lesion, whereas the patients who have this mutant PPP2R1A mutant, and most of them goes down along with this AKT mutant, and some of this PPP2R1A and AKT wild type also goes down, but majority of them goes down or belongs to the mutant conditions. So this together indicates that there might be possibilities that PPP2R1A mutants will have improved effect of the ICB, that is immune checkpoint blockade treatment, on the patients who are affected with this OCCC, that is ovarian clear cell carcinoma. That's all for this video. We will discuss further results in our subsequent video. So if you need to understand more about these experiments in detail, you can go through the reference that has been provided in the description. Thank you for your time.